Hello, we are back with another, what's this called? DNA talk, and we are going to talk about chromatin this time. Let me just check something. Here it is. DNA packaging. All right. And there should be there. But yes, okay. So chromatin is a complex of DNA and associated proteins that make up the structure of chromosomes in a eukaryotic cell. Okay? And changes in the chromatin structure, they are closely linked to regulation of gene transcription. We talked about that earlier. That would be the histones, acetylation, methylation, and etc. We're going to dive deep into that one today. And modifications to the chromatin, such as addition of chemical groups to histones or alterations in DNA accessibility, would play a crucial role in turning the genes on or off, and these dynamic changes are fundamental for controlling gene expression, cell differentiation, and responses to environmental signals. And we have here a photo of a eukaryotic cell nucleus. And it's essentially an electron micrograph of the panel. Let me just check something. Yeah. It's an electron micrograph. You have the nucleus in the middle. That's the central compartment which contains the cell's genetic material. Guess what's that? It's the DNA. And you have the nuclear envelope. It's the double membrane structure surrounding the nucleus. It has an inner and outer membrane, and it consists of nuclear pores that regulate the entry and exit of materials. You also have the nucleolus, which is a very dense region within the nucleus where you have rRNA is being synthesized and ribosome units are assembled. The nucleolus might be denser than your average main character in anime, especially since you need a lot of rRNAs being synthesized over there. And then we have the chromatin. The chromatin is a complex of DNA and it is associated, it's DNA and it's associated proteins, all right? And with that, it can exist in a condensed or decondensed state, aka heterochromatin and euchromatin. Heterochromatin is the condensed state, meanwhile euchromatin is the relaxed state. And shifting between those forms would be related to gene expression. You also have the centrosome and the microtubules, which is important for cell division and maintaining the cell shape. And these structures help organize the microtubules that form part of the cytoskeleton. For the cytoskeleton part, we will talk about that in a future one. And then you have the nuclear pores. Those are just protein complexes that control the transport of molecules between the nucleus and the cytoplasm. Pores, essentially, they're holes. Okay? Let me just grab water 1 second. I'm back. I have the water now. Wait. Okay. Chromosomes in eukaryotic cells display different structural forms, and that depends on the cell state. In a dividing cell, the chromosomes are highly condensed and visible because they're very, very condensed, so it's easier to see them. Meanwhile, if they are non-dividing, it's more relaxed and it's not really visible because it's in an extended state. So, and that difference in chromatin organization is significant because the condensed form during cell division is the one used during segregation, okay? Meanwhile, extended version or the heterochromatin would be useful for the euchromatin would be useful for gene expression and DNA replication in the non-dividing cells. So again, heterochromatin, the condensed one, is usually during the segregation. Meanwhile, euchromatin would be most likely for like gene expression because the different transcription factors can come in. All right, now let's talk about packaging of DNA. Well, you begin with the DNA double helix, the very common picture that you know. It's about 2 nanometers in diameter. And then the DNA wraps around the histone proteins, so they circle around the circles. And then, after the DNA double helix wraps around the histone proteins, that one is now called a chromatin. Wait, let's just double check again. DNA wraps around histone, which would form nucleosomes. Okay, that's the beads on a string. The nucleosomes would then coil into a chromatin fiber, which is around 30 nanometers. Okay, so DNA wraps around histones, which would form nucleosomes that's around 11 nanometers thick. The nucleosomes would then coil into a 30 nanometer thick chromatin fiber. The chromatin fiber would condense even more, which would form larger loops, and the thickness would be around 300 nanometers. And to prepare for cell division, it would super duper condense to around 700 nanometers. And then after that, you then get the chromosome, which is around 1,400 nanometers. Okay, that's during mitosis. It's approximately 10,000 times shorter than its normal length. Than its extended length, And this hierarchical packaging would ensure that long DNA molecules could fit inside the nucleus, especially during the segregation part of mitosis. Okay, so again, you have the DNA double helix wrapping around the histones. So the 2 nanometer one would then become like around 11 nanometers, and then the histone. One moment. DNA double helix would wrap around the histones, forming nucleosomes. That's around 11 nanometers. The nucleosomes would then coil into a 30 nanometer chromatin. Chromatin would then further coil into loops. The loops would then coil once more until they become 1,400 nanometer chromosomes. Okay. And the chromosome, that would be, we have 23 of them normally. And that has a centromere in the middle. Okay. So now, nucleosome structure. Nucleosomes are basic structural units of chromatin. Why? Because that's the beads on a string. And it consists of DNA wrapped around a core of histone proteins. And this is important because this core histone complex includes 2 copies, each of histones 2A, 2B, 3, and 4. H2A, H2B, H3, and H4. 2 copies of each, you get an octamer. And the DNA around this core histone complex is around 147 base pairs long. Okay? And you have an additional DNA linking one nucleosome to the next, and that's why you have the beads on a string appearance. So, nucleuses can digest the linker DNA between the nucleosomes, and that would release the nucleosome core particle. And when you add salt, a lot of salt, the nucleosome structure will dissociate and release the DNA from the histone. And now you see the inside its core, it's the 2A, 2B, 3, and 4. 2A, 2B, 3, and 4. And so again, you wrap around, and there's the linker DNA in the middle, and then wrap around again. Then what are the primary structures of histone? Histones? Well, it has a histone fold region and a flexible and terminal tail. The histone fold region is highly conserved, and it forms a three-dimensional structure essential for the histone's function. And each histone protein, H2A, 2B, 3, and 4, they have structural differences. But, they differ slightly. They have similar structures, but they differ in their amino acid sequence. So that's why in some pictures there are different colors to represent them. How are the histones like dimered or tetramered like that? Well, histones essentially, they are again proteins that help the DNA package into nucleosomes. And before they form a complete nucleosome core, they first exist as dimers and tetramers. So first, histones 3 and 4 would stick together. And then it would call in another histone 3 and 4. So now we have a double 3, 4, 3, 4. And then in another side, you have the 2A and 2B sticking. And then it would then attach itself to another H2A, 2B. So now that's a dimer. From a dimer into a tetramer. And then the two tetramers now would just, and now they are, an octamer. You get your histone octamer. So that's how it forms. Again, how is a nucleosome formed? Well, first you have histone 3, 4, 3. Let's say histone 3 and 4 sticks together, and then another histone 3 and 4 stick together. Those are now dimers and dimers. The two dimers stick. Now you have a tetramer. In another universe, histone 2A and 2B sticks together. Another 2A and 2B sticks together. Mix those. Histone 2. You have the tetramer now. The two tetramers, bam, together, and now you have your octamer, the histone octamer, with a lot of the end tails like wrapping around histone. And now next, histone tails are flexible, and some unstructured N-terminal extensions of histone proteins protrude from the histone core, like the 2A tail, 2B tail, 3 tail, 4 tail. And the tails are crucial in the regulation of chromatin structure. Usually, they are edited or manipulated through post-translational modifications like acetylation, methylation, and phosphorylation. So those would just alter how histones interact with DNA and proteins, and thereby influencing the condensation and accessibility of chromatin for processes like transcription, replication, and repair. Okay. Histone tails, which are flexible regions of histone proteins, interact with adjacent nucleosomes to help regulate the structure and dynamics of chromatin, and these interactions play a crucial role in the compaction and organization of DNA in the nucleus. Histone. So earlier, we only talked about histone 3, 4, 2A, and 2B, but what happened to the other numbers? Well, you have histone 1 now, histone 1 would interact with the tails of histone 4s. Okay, and in this process, histone 1 binds to the tails of 2A, 2B3, and 4, and that's now it will contribute to the structural changes and stabilization of chromatin. And this interaction is essential for further compaction of the DNA. So again, the N-terminal tail of histone H1 interacts with the linker DNA and the histone tails, and it will lead to like regulation of it. Histone 1 is just there. It will interact with the others. And unwrapping and rewrapping of nucleosomes would play a key role in regulating protein binding to DNA. When the nucleosome is in a very wrapped state, it prevents proteins from accessing the DNA. However, the nucleosome can transiently unwrap, with exposing the DNA for a very, very short time, 10 to 15 milliseconds. And this unwrapped state would allow the specific DNA binding proteins to bind to their target sites on the exposed DNA. And once the protein binds, the nucleosome quickly rewraps around the DNA, and that's why you can do gene expression with this. Okay? Okay, good. Now you have chromatin remodeling complexes. What are they? Well, I'm gonna talk about them now. They are ATP dependent, and they play a crucial role in regulating chromatin structure by catalyzing nucleosome sliding. Sliding, what's that? Well, the chromatin remodeling complex binds to the DNA. It would hydrolyze ATP to ADP, and then after that, this energy is then used to move nucleosomes along the DNA. Okay? So this makes the DNA more or less accessible, whether the chromatin is condensed or decondensed. And when you have repeated rounds of nucleosome sliding, you could regulate the accessibility of specific DNA regions, which would impact the gene expression. And in its decondensed form, again, it's more accessible. So again, what's the function of a chromatin remodeling complex? It will assist with the nucleosome sliding, and which might expose specific genes or areas in the DNA. It needs ATP though. Okay. CRC, chromatin remodeling complex. So another term for its function is nucleosome displacement. And this one's just a different picture. You have the octamer being displaced enough, and then it's now a little bit open, and RNA polymerase can bind, and boom. Next is chromatin structure and gene transcription. In chromatin, the 30 nanometer fiber structure is folded into looped domains. And those are critical for gene regulation because within the looped regions, histone modifying enzymes, chromatin remodeling complexes, and RNA polymers interact. So just remember that when they open up, the different things can come in, and then boom, you can have more higher transcription. Or if you just make it like more condensed, then nope, no transcription. DNA Swan, what is it? DNA Swan is DNA Swan hypersensitive sites. They are located approximately 50 kb 50 kb 5 prime and 20 kb 3 prime of the beta globin gene in developing human erythrocytes. I think this is just a very specific example, and that plays a key role in the beta globin expression. So for transgenic mice to express the human beta globin gene at high levels, the trans gene must include not only the beta globin gene itself, but also very hypersensitive sites along its promoter proximal elements. So when the hypersensitive sites are present, the gene is expressed at very high levels. Okay? Again, the DNase 1 hypersensitive sites are available there, and if they are not there, expression is so much lower. So, I mean, yeah, you get it. If the site is hypersensitive, then transcription would be higher. And the expression of the beta globin gene family is regulated by a distal locus control region, LCR, upstream. The gene cluster and the locus control region plays a crucial role in the regulation of these genes. Okay? And during fetal development, the synthesis of the gamma G and gamma A globins is high, but after birth, synthesis declines, while the synthesis of the delta and beta globins increase. And this shift from fetal to adult globin production is very tightly controlled with the LCR. So that's crucial in development. Again, the distal control region is responsible for that. All right, now recruitment of chromatin remodeling proteins and histone acetylases during the initiation of transcription can occur, but it happens. So during the initiation of transcription, an activator protein could bind to an enhancer region, and this serves as a binding site for the activator protein. The enhancer region, because there's like enhancer region, that's the part in the gene, and then an activator protein binds to it. It will dock. That's its parking lot. And this interaction facilitates the recruitment of other stuff necessary for transcription, including general transcription factors, RNA polymerase, mediator, chromatin remodeling complex, and histone acetylases. And they assemble at the Tata box. The mediator complex and other stuff would then go there. And that would just allow the transcription into R and A. Just to review, how does DNA transcription occur? You have, okay, I was about to say the stuff for replication. Not that. Okay. For transcription, just going, I'm looking for the specific one. Not this. One moment. Tata box. Okay, then. For transcription, you have TF2D, which has the, okay, TF2D, which has the TBP, goes to Tata box. It calls in 3 stuff, E, H, and the RNA pol 2. Pol 2 would have F, TF2F, with a CTD. Let's put the sticky note here. So again, how does transcription occur? For initiation, you have the TF2D, which has the Tata binding protein area. It would go to the Tata box, and then 3 or 4 stuff is called TF2E, TF2H, and then RNA pol 2, which has TF transcription factor 2F, and which has the CTD, the something terminal, the C terminal domain. That's also called. And then after that one, you just have helicase, and then you have the RNA pol, which can do its thing. Lysine, going back to acetylases and etc. Lysine residues on histones can undergo either acetylation or methylation. And these modifications are competing reactions. In acetylation, an acetyl group such as, it's like COCH3. It's added to the lysine residue, and you can get an acetyl lysine. This would, if you acetylate, it relaxes. It relaxes the chromatin structure. Again, if you acetylate, it relaxes the chromatin structure, aka more accessible for the gene regulation. And if you methylate, methyl is added, it would usually compact it. So again, acetyl A is more, if you acetylate, it's more expanded now, decondensed, relaxed. Meanwhile, methylate, it would lead to tightening of it. And that's because there was a good one earlier. Is it this one? No. We talked about it in a previous slide. Not the homologous recombination. Strand direct repair, no. What was it? Transcriptional control. Thyroid, nope. Gotta find that part. Because it explained it much better. Cerevisiae, no. Let me just find that picture. A moment. Not the promoters. Lol, it's the amanitin sensitivity. RNA pol, reminder, RNA pol 1 is more insensitive. It's very insensitive. RNA pol 2 strongly inhibited. Pol 3, it happens if it's like higher. Can't find it. It was about DNA. Obviously about DNA. Okay, it's fine. Wait, was it just super recent? All the things are mixing up. Alright. So again, normally it is positive or negative, right? So we know that the histone core is more positive, and then you have the phosphates on the DNA. It's more negative. If you acetylate it, that will, an acetylysine. So that will lead to a more relaxed chromatin structure, because it will lead to, I think it was like the neutralizing part. Yeah, the histone. Oh, it's the T3 part. The deacetylates, the HDAC and HAT. I remember. Where was it? No. Here. There you go. Yeah. So if you acetylate it, the acetylation neutralizes the positive charge on the lysine residues, which would then lead to weakening the interaction of between the histone core and the DNA. Good. Now, if you phosphorylate, this is another thing that you gotta remember. If you phosphorylate, you mostly with serine phosphorylation. So it's a modification that occurs on histones where you add a phosphate group on the hydroxyl. Phosphate is a little bit negative, and this could lead to activation or repression depending on the context. So it's not very clear cut. So you have post-synthetic modification of histones. Again, it's 2A, 2B, 3, and 4. And these modifications primarily occur on the N-terminal tails of the histones. You can either methylate, phosphorylate, acetylate, and ubiquitinate. A lot of things you can do with it. And that, it just depends on the different stuff that happens. So, if you methylate, this one, okay. If you methylate on lysine 4, what? Okay, acetylating on lysine 9 and methylating on lysine 4 would lead to gene expression. This is just specific. But methylate the gene lysine 9, it would be silencing. Acetylate, most here, I was gonna, okay. Methylate, it just depends. There are some weird stuff. But generally, acetylating would lead to gene expression. But methylation is specific. That one is a depend. Like it depends. Okay. Now let's. Oh, this is a good one. It's the camp kind CAMP. So when a ligand binds to a receptor in the plasma membrane, it would activate the receptor, which would activate the G protein complex in the cell. G protein complex would trigger and the enzyme adenylyl cyclase, and the adenylyl cyclase would catalyze the conversion of ATP to CAMP. And the release in CAMP would activate PKA, because the CAMP would then attach to the PK. There's like, the PKA has very specific areas for it where CAMP can attach to. So once that happens, the PKA subunits would detach from that, and then it can travel to inside the nucleus. And then with that one, they phosphorylate Krebs. They phosphorylate Krebs using ATP to ADP. If Krebs is phosphorylated, okay, focus here. If Krebs is phosphorylated, they recruit Krebs binding protein. Okay, Krebs calls CBP. Krebs binding protein has an activation domain that would initiate the transcription. And that is at the CRE site, CAMP response element, on a specific gene. So that's how the signaling cascade goes from the ligand to the gene transcription, gene regulation. This we will talk a lot during the GPCR and the tyrosine something. Cell signaling. Oh, and going back to the absence or presence of T3. So this one, we have, okay, if there is no T3 and TR, or T3 would just bind to RXR. RXR would call in, there's a different picture that I like. 2 seconds. There you go. T3. Yeah. If you don't have the thyroid hormone, T3, the thyroid receptor would just bind to the RXR. It would call in 3 things: NCORE, SMORT, and HDA. NCORE is nuclear receptor co-repressor. So TR, RXR, NCORE, silencing mediator for retinoic acid, SMRT, and thyroid hormone receptor. And the HDA, histone deacetylase. If you deacetylate it, then you do not have transcription, because if you deacetylate, then you remove the acetyls. The DNA can just bind more tightly to the histone core, so it will just close up. So again, without the thyroid, you would just have the TR would bind to RXR. You have the NCORE, SMORT, and HDA. But then if you have the thyroid, then TR to T3, it would call in RXR, and the friends that they will call would be drip trap and SRCHAT. What's drip trap? That's the vitamin D receptor interacting protein. And then SRC would be steroid receptor co-activator. You have the histone acetyltransferases. It would acetylate it, and then it would neutralize the charges on the lysine, the acetyls, and then it would just loosen up the bond or attraction between DNA and your histone core. And so that's why in the presence of the thyroid, it can now the transcription is de-repressed. It can happen. Okay. Transcription. This one is also another talk about cadmium and MTF. Essentially, if you bind the cadmium with metal, so the process begins with the binding of metal ions such as zinc and cadmium or hydrogen peroxide, and that will lead to a change in conformation with the MTF fingers, and that would phosphorylation and dephosphorylation of MTF1 would regulate the activity. It's just very specific stuff. So what is this one? Writer and reader complexes and gene transcription. In gene transcription regulation, protein molecules bind to specific histone modifications on the nucleosome, and that would result in a covalent modification of the histone tail. That's commonly referred to as a mark, and scaffold proteins interact with the mark. So again, during the gene transcription regulation, some protein molecules bind to specific histone modifications on the nucleosome. And then that would result in a modification of the tail, such as the mark. Some proteins interact with the mark, forming a code reader complex. This code reader complex binds to it binds to the modified histones and attracts other components. And the assembled complex attaches to other components in the nucleus, influencing gene expression, gene silencing, and other stuff. Crucial detail would be the code reader complex binds and attracts the other components. In the human gene or human interferon gene promoter, a gene activator protein would bind to the promoter region, specifically near the Tata box, and that's essential for transcription initiation. So again, you have the human, that's a human. There's an interferon gene promoter at the start site. The gene activator protein binds to the promoter region near the Tata box, and that's again, it's essential for initiation. Histone acetyltransferases also comes in, and the enzyme would modify histone for the H4K8 and H3K9 by adding acetyl groups. Adding acetyl groups would loosen the chromatin structure, and then you can have access to the DNA and allowing the transcription machinery to happen. So again, you have the gene activator protein binding to the area near the Tata box, that's the promoter region. Histone HAT also comes in, and it would acetylate the H4K8 and H3K9. Add them, and boom, you can have acetylation. And then acetylation and phosphorylation of H3 is histones 3 play an essential role in regulating it. Yeah, it's just repeating the thing. Acetylated, further acetylation of H3 would just lead to more DNA. After this, the binding of the chromatin remodeling complex is a key step in transcription initiation. So yeah, after you modify H3K9, so all in H3, K9, S10, and K14, that would form the histone core for initiation. And then the chromatin remodeling complex would then come in, and additionally, you have the TF2D binding. That's the start of everything. So the main part is histone 3 is really edited in H3 before transcription. So you acetylate H3 where? At S10 and K9. You have histone kinase coming in. What are some possible ways or possible pathway for transcription activation? Well, it begins when a gene activator protein binds to the chromatin, and then it would recruit the chromatin remodeling complex. So again, you have an activator protein binding to chromatin. Chromatin remodeling complex is recruited. After that, histone modification enzymes. The histone is now modified. Then you have other activator proteins such as mediator, transcription factors, and gene activation until you have the transcription initiation. Okay, and just last 2 points would be the methylation, cytosine methylation. That would involve the addition of a methyl group to the carbon 5 position of the ring, the cytosine ring. Wait, one prior to transcription and initiation. Okay, so when you add a methyl group to the carbon 5 position of the cytosine ring, when this methylation occurs, the cytosine is now a 5-methyl cytosine. Okay? And then, if that happens, well, yeah, that's true. If that happens, you have a 5-methyl cytosine now. And methylation of cytosine in DNA, and well, that would just lead to different stuff. An increase in 5-methylcytidine followed by what is a G? MCG. It would lead to repression of gene transcription. So if you methylate it, so methylation of cytosine in DNA involves addition of methyl group to the 5 position of cytosine, right? If you have a 5-methyl cytosine, 5-MC, adding it by, and then if it is followed by a guanine, so you have an MCG, that will lead to repression of gene transcription. If you increase methylation of cytosine and it is a 5-MCG now, that would lead to lower transcription because methylated cytosine would serve as a binding site to recruit MSyn3 and etc., which has histone deacetylase activity, and that's why it could lead to repression, ultimately leading to decreased gene transcription. Just as a review, some basic conceptual questions. What is the role of chromatin structure in regulating gene expression? Well, the chromatin, it would be so to regulate gene expression, you would control DNA accessibility in it. And in a condensed form, heterochromatin, the chromatin is less accessible to the transcription machinery, resulting in gene silencing. In a relaxed form, the euchromatin is more accessible, allowing the transcription factors and RNA polymerase to bind to DNA and initiate the transcription. How does histone modification affect chromatin structure and transcriptional activity? So you have histone modifications such as acetylation, methylation, and phosphorylation. Those would influence the chromatin structure by altering the interactions between histones and the DNA. And that's because acetylation typically neutralizes the positive charge on histones, which would lead to a more open structure. And then methylation would, specifically at around the lysine residues, if you methylate, it would lead to gene silencing because it would promote the condensation. Okay. I think next is if I wanted to design an experiment to assess the accessibility of chromatin for transcription factors in a given gene region, maybe chromatin immunoprecipitation such as CHIP, followed by qPCR or ChIP-seq, can be used to determine whether specific transcription factors would bind to a gene region. And in this experiment, the cells can be treated with histone modification inhibitors. Because if you do that, the binding of transcription factors to specific regions can be measured, and the difference in the binding, the presence or absence of inhibitors would show how histone modifications affect it. All right. And then how do I determine if when histone modifications are associated with active gene transcription? ChIP-seq would be good, especially for active transcription, because you have specific antibodies against the acetylated or methylated histones, and that can be used to immunoprecipitate them. And you can just use gene sequencing to identify where the modifications occur along the genome. How would you investigate the role of chromatin remodeling complexes in transcription activation? Some reporter gene assays can be used to study chromatin remodeling, and cells are transfected with a reporter gene to check that out. There's also the chromatin sliding. Nucleosome sliding, we mentioned that. Gene silencing, that's for the methylation. Histone acetyltransferases and HDAC also works. And then the metal ions, if you bind the zinc, cadmium, or H2O2, MTF1, you have a conformational change that would lead to it binding to MRE. And then if you have the MRE, you activate the transcription. All right. So I think for the chromatin topic, that is all for that one. Thank you for watching. Bye.